तो वेलकम यू टू ऑल। आज हम पढ़ेंगे यूनिट 3 का सेल स्ट्रक्चर एंड फंक्शन का चैप्टर 8, क्लास 11 बायोलॉजी। सेल द यूनिट ऑफ लाइफ। इसमें हम पढ़ेंगे व्हाट इज़ अ सेल, सेल थ्योरी, ओवरव्यू ऑफ सेल, प्रोकैरियोटिक सेल, यूकैरियोटिक सेल। यहाँ एक बहुत सुंदर डायग्राम दे रखा है सेल का। G1 फेज, G0 फेज, S फेज, G2 और M फेज। M फेज में यानी माइटोसिस में आते हैं साइटोकाइनेसिस, टीलोफेज, एनाफेज, मेटाफेज, प्रोफेज। चलिए शुरू करते हैं। When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living? Or what is that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, multicellular organism. What is a cell? Unicellular organism capable of independent existence and performing the essential function of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structure and functional unit of all living organism. The fundamental है. Anthony von Leeuwenhoek first saw and described a living cell. Robert Brown later discovered the nucleus. The invention of the microscope and an improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Sichelden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodor Sachman, 1839, a German zoologist, studied different types of cells, animal cells, and reported that cells had thin outer layer which is today known as plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cell. On the basis of this, Sachman proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. So, who? किसने किया था ये? सचवान. Sheldon and Sachman together formulated the cell theory. This theory, however, did not explain as how new cells were formed. Rudolf Virchow first explained the cell divide. New cells are formed from pre-existing cells, omnis cellula e cellula. It was Rudolf Virchow. He modified the hypothesis of Sheldon and Sachman to give the cell theory a final shape. Cell theory as understood today is all living organisms composed of cells and products of cells. All cells arise from pre-existing cells. An overview of cell. You have earlier observed cell in an onion peel or human chick cells under a microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as an outer boundary, and just within it is a cell membrane. The cells of the human chick have an outer membrane as a delimited structure of the cell. Inside each cell is a test membrane bound structure called nucleus. This nucleus contains the chromosome, which in turn contains the genetic material DNA. Cells that have membrane bound nuclei are called eukaryotics, whereas cells that lack a membrane bound nucleus are prokaryotic. In both pro and eukaryotic cells, a semi-fluid matter called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activity in both the plants and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Beside the nucleus, the eukaryotic cells have another membrane-bound distinct structure called organelles, like the Endoplasmic Reticulum, the Golgi complex, Lysosomes, Mitochondria, Microbodies, and Vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membranous bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plant and mitochondria and or rough ER. Animal cells contain another non-membrane bound organelle called centrosome, which help in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasmas, the smallest cells are only 0.3 meter in length, while bacteria could be 3 to 5 meter. The largest isolated living cell, single cell, is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7.0 meter in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboidal, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Nerve cell देख लेंगे, a tracheate elongated देख लीजिए, basophil cell round and oval, white blood RBC, columnar epithelial cell long and narrow. Prokaryotic cells are represented by bacteria, Golgi algae, Mycoplasma, and PPLO, pleuro pneumonia-like organism. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may even vary greatly in shape and size. The four basic shapes of bacteria are Bacillus, Coccus, Vibrio, and Spirillum. Organization of the prokaryotic cell is fundamentally similar even though prokaryotes exhibit a wide variety of shapes and function. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The semi-fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition to the genomic DNA, the single chromosome, circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmid. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryote cell except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially infoldings of cell membrane. As you can see, typical bacteria 1 to 2 meter, PPLO, viruses, typical eukaryotic cell 10 to 20 meter. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx followed by the cell wall and then the plasma membrane. Although each layer of envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelope and the manner in which they respond to the staining procedure developed by Gram, which those that take up the Gram stain are Gram positive and those do not are called Gram negative. Glycocalyx differ in composition and thickness. Glycocalyx differ in composition and thickness among different bacteria. It could be a loose sheath called the similar in some, while in others it may be a thick and tough called the capsule. The cell wall determines the shape of the cell and provide a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interact with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membrane structure the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. They help in cell wall formation, DNA replication, and distribution to daughter cell. They also help in respiration, secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membrane extensions into the cytoplasm called chromatophores which contain pigment. Bacterial cells may be motile and non-motile. If motile, they have thin filamentous extension from the cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Beside flagella, pili and fimbria are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structure made up of special protein. The fimbria are smaller bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Ribosome and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. There are about 15 nanometer by 20 nanometer in size and are made of two subunits 50S and 30S, which when present together form 70S prokaryotic ribosomes. Ribosome are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. The ribosome of a polysome translates the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophysine granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cell. The eukaryotes include all the protists, animal, plants, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different, as the former possess cell walls, plastid, and a large central vacuole, which are absent in animal cells. On the other hand, animal cells have centrioles, which are absent in almost all plant cells. As you can see the diagram, this is rough endoplasmic reticulum, lysosome, smooth, plasmodesmata, microtubule, Golgi Apparatus, nucleolus, nuclear envelope, cell wall, mitochondria, perixome, and this is the animal cell, leg cell wall. The detailed cell membrane. The detailed structure of the cell membrane was studied only after the advent of the electron microscope of 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cell, enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to the phospholipid, membrane also contains cholesterols. Later biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipids varies considerably in different cell types. In humans, being membrane of the erythrocyte has approximately 52% protein and 40% lipid. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral protein lie on the surface of membrane while the interior proteins are partially or totally buried in the membrane. Peripheral, you can see sugar, phospholipid bilayer, integral protein, fluid mosaic model of plasma membrane. An improved model of structure of cell membrane was proposed by Singer and Nicholson, widely accepted as the fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as fluidity. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation of intracellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the cell membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient from higher concentration to the lower. The fluid nature of the membrane is also important from the point of view of junctions like cell growth, formation of intracellular junctions, secretion, endocytosis, cell division. One of the most important functions of this membrane is the transport of molecules across it. The membrane is selectively permeable to some molecule present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower to the higher. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example, Na plus K plus pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectin, and protein. The cell wall of a young plant, the primary wall, is capable of growth, which gradually diminishes as the mast cell mature, and secondary wall is formed on the inner, towards membrane side of the cell. The middle lamella is a layer primarily of calcium pectate which hold or glues the different neighboring cells together. The cell wall and middle lamella may be transferred by plasmodesmata which connect the cytoplasm neighboring cell. Endomembrane system. While each of the membrane granules is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Since the function of the mitochondria, chloroplast and Peroxisomes are not coordinated with our components. These are not considered part of the endomembrane system. Endoplasmic reticulum (ER). Electron microscope studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum (ER). Hence, ER divides the intercellular spaces into two distinct compartments, luminar inside ER and extraluminal cytoplasmal compartment. ER often show ribosome attached to the outer surface. The endoplasmic reticulum bearing ribosome on the surface is called rough. In the absence of ribosome, they appear smooth and are known as smooth endoplasmic, as you can see. ARIA is frequently observed in the cell activity involving protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cell, lipid-like steroidal hormones are synthesized in SCR. Golgi Apparatus. Camillo Golgi, 1898, first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat, dish-shaped sacs or cilia of 0.5 nm to 1.0 nm diameter. These are stacked to parallel to each other. Varied number of cisternia present in the Golgi complex. The Golgi cisternia are concentrically arranged near the nucleus with distinct convexes or the forming phase and concave trans or the maturing phase. The cis and trans phase of the organelle entirely different but interconnected. The Golgi apparatus principally performs the function of packing materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packed in the form of vesicle from the ER fuse with the cis phase of the Golgi apparatus and moves towards the maturing phase. This explains why the Golgi apparatus remain in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternia of the Golgi apparatus before they are released from its transphase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Lysosomes. These are membrane-bound vascular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzyme, hydrolases, lipase, protease, carbohydrates, optimally activate the acidic pH. These enzymes are capable of digesting carbohydrate, protein, lipid, and nucleic acids. Vacuoles. Vacuoles is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoblast. In plant cell, vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. In terms of shapes and size also, considerable degree of variability is observed. Typically, it is saucer-shaped or cylindrical, having a diameter of 0.2 to 1.0 mm, average 0.5 mm, and length 1.0 to 4.1 mm. Each mitochondria is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments. That is the outer compartment and the inner compartment. As you can see in the diagram, matrix, cilia, inner, outer. The inner compartment is filled with dense homogeneous substance called the matrix. The outer membrane forms a continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the single cilia, towards the matrix. The cilia increases the surface area. The two membranes have their own specific enzyme associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouses of the cell. The matrix also possesses singular circular DNA molecule, a few RNA molecules, ribosomes, and the component required for the synthesis of proteins. The mitochondria divide by fission. Plastid. Plastid are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, imparting specific colors to the plant. Based on the type of pigment, plastic can be classified into chloroplast, chromoplast, and leucoplast. The chloroplasts contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophyll, and others are present. This gives a part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastic, varied shapes and sizes with stored micronutrients. Amyloplasts store carbohydrate, starch, example potato. Elioplast. Elioplast store oil and fat, whereas the aleuroplast store proteins. Majority of the chloroplasts of the green plants are found in the mesophyll cell of the leaves. These are lens-shaped ovules, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 mm and width 2 to 4 mm. Their number varies from 1 per cell of the Chlamydomonas or green algae to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound of the two. The inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called stroma. A number of organized flattened membrane sacs called thylakoids are present in the stroma. Thylakoids are arranged in stacks like the piles of coins called grana, singular granum, or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamella connecting the thylakoids of the different grana. The membrane of thylakoid encloses a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrate and protein. It also contains small, double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, 70s, than the cytoplasmic ribosome, 80s. Ribosomes. Ribosomes are the granular structure first observed under the electron microscope as dense particles by George Palade, 1953. They are composed of ribonucleic acid and protein and are not surrounded by any membrane. The eukaryotic ribosomes are 80s, while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller. The two subunits of 80s are 60 and 40s, while that of 70s ribosome are 50s and 30s. Here S, Swedberg's unit, stands for the sedimentation coefficient. It is used indirectly a measure of density and size. Both 70s and 80s ribosomes are composed of two subunits. Cytoskeleton, an elaborate network of filamentous proteinish structure consisting of microtubules, microfilaments, An intermediate filaments present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton is cell line involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. As you can see in the diagram, plasma membrane, central sheath, peripheral microtubules, interdoublet bridge, central microtubule, radial spoke, cilia and flagella. Cilia, singular cilium, flagella, singular flagellum, are hair-like outgrowth of the cell membrane. The cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella comparatively longer, responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. Electron microscope study of cilium or the flagellum showed that they are covered with plasma membrane. Their core, called the exonium, possess a number of microtubules running parallel to the long axis. Exonium usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of exonymal microtubules is referred to as a 9+2 array. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. Centrosome and Centrioles. Centrosome is an organelle usually containing two cylindrical structure called centrioles. They are surrounded by amorphous pericentriolar material. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin proteins. Each of the peripheral fibrils is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of proteins. Centrioles form the basal body of cilia, flagella, and spindle fibers that give rise to spindle apparatus during cell division in animal cells. Nucleus. Nucleus is cell organelle was first described by Robert Brown as early 1831. Later, the material of the nucleus standard by the basic die was given the name chromatin by Fleming. So, chromatin नाम किसने दिया? Fleming ने. The interphase nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli, singular nucleolus. Electron microscope has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remain continuous with the endoplasmic reticulum and also be a ribosome on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules take place in both direction between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation number of nuclei are also frequently observed. Some mature cell even lack nucleus, example erythrocytes of many mammals and sieve tube cells of vascular plants. इसमें nucleus नहीं होता है. Would you consider these cells as living? So erythrocytes in RBCs और sieve tubes के जो cells होते हैं उनमें nucleus नहीं होता है. The nuclear matrix of the nucleoplasm contains nucleus and chromatin. The nuclei is spherical structure present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nuclei are present in cells actively carrying out protein synthesis. You may recall that the interface nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contain DNA and some basic protein called histone, some non-histone proteins, and also RNA. A single human cell has approx 2-meter long thread of DNA distributed among its 46 23 pairs chromosomes. You will study more about it in class 12th. Each chromosome, visible only in dividing cells, essentially has a primary construction of the centromere, on the sides of which dish-shaped structure called chromatochores are present. Chromatomere hold two chromatids of a chromosome. Based on the position of the centromere, chromosome can be classified into four types: the metacentric, subcentric, acrocentric, telocentric. In metacentric, chromosome has middle centromere forming two equal arms of the central chromosome. Middle में है centromere. Submatrix chromosome has centromere slightly away from the middle, resulting into एक short, एक बड़ा, एक छोटा. In acrocentric, close to one end. इसमें this one acrocentric. And telocentric में extreme one end पे होता है centromere. सो मेटासेंट्रिक यानी बराबर चीज़ें मिडल में है. टीलोसेंट्रिक बिल्कुल ही एक एज पे है, बीच में आता है. सब मेटासेंट्रिक थोड़ा ऊपर, एंड एक्रोसेंट्रिक आफ्टर द मेटासेंट्रिक, सब मेटाइम. Sometimes few chromosomes have non-staining secondary constructions at a constant location. They gives the parent small fragments for satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Okay, this is a summary. Kindly read it and let me know if you have any doubts. Also the exercise. Do like, share and subscribe so that I can make more such content. Thank you so much for watching.